Repeat here. Okay. Good evening, everybody. Today I am going to discuss about the brief note or a brief explanation on the exoplasmic transport. Exoplasmic transport is the transport or the transfer of substances between the cell body and the axon terminal. It is called as exoplasmic transport. So, whatever the solutes are present in the cell body, these solutes they are transported from the axon to the from the cell body to the axon. So here there are various proteins, organelles and other cellular substances required for the development of growth and maintenance of neuron and transported mainly along the length of axon. Abolished by application of colchicine, dinitrophena, azide, cyanates and prolonged anoxia. Colchicine disrupts the movement of microtubules. So depending upon the direction of transport, we can classify exoplasmic transport into three. One is anterograde transport, retrograde transport, and transneuronal transport. Anterograde, retrograde, and transneuronal transport. Anterograde, retrograde, and transneuronal transport. Anterograde transport, the transport of materials from the cell body towards the axon terminal. So, starting from the cell body, all the solutes they are transported towards the axon terminal. That type of transport is called as a anterograde transport. All the neurotransmitters, neurotransmitters, they belong to this category. So there are different neurotransmitters. All the neurotransmitters belong to this category only. This type of transport only. The best example is it is a fast and slow. Next, fast exoplasmic transport, the speed about 400 millimeters per day by kinesin. Kinesin material transported organelles, vesicles, and membrane glycoproteins. And it is a neurofilaments, actin and microtubules take part in the slow exoplasmic transport. Retrograde is in reverse direction from the axon terminal to the cell body. If the solutes are transported from the axon terminal to the cell body, that is called retrograde, brought about by a protein called dynein. Dynein is a protein, carrier protein, and it is mapped by horseradish peroxidase. What are the examples of retrograde transport? The transport of viruses, chickenpox virus, rabies virus travels from axon ending to the motor neuron to the spinal cord and then brain where it multiplies. Transport of toxins, transport of nerve growth factors, reuptake of synaptic transmitters, all these are the best examples of the retrograde transport. And what is meant by the trophic substances like transport? Trophic substances like nerve growth factors are transported across the synapse to the presynaptic membrane of another neuron. This is called as a transneuronal transport. Transneuronal transport. This helps in maintenance of synaptic contacts. So, synapse means it is a junction between the two different neurons. So, what are the functions of neuron? The cell body, anatomical integrity of axon, maintains the functional and anatomical integrity of the axon. The synaptic transmitters that are synthesized in the cell granules of the cell body and are transported to the axon terminal. Dendrites, they receive impulses. Axon hillock internal segment site where the action potential is generated. The metabolism is a nerve for a considerable period in the absence of oxygen. If water supply is sufficient, pyruvic acid is formed, and if water supply is insufficient, lactic acid accumulates. So during activity, hydrolysis of OTP and creatine phosphate supply energy. Vitamin B1 is essential for vitamin B1 is vitamin B1 is essential for oxidation of pyruvic acid and lactic acid in the neurons. So, what are the growth factors? Various factors affect the neuronal development. Are called neurotrophins. Ciliary neurotrophic factor, glial cell line derived neurotrophic factor, leukemia inhibitory factor, FGF, platelet derived growth factor, TGF, IGF, all these are the different neurotrophins. These are trophic proteins to the neurons. They promote nerve growth and survival. So, what is the main function of neurotrophins? Neurotrophins are the proteins to the neurons, trophic proteins to the neurons, as they promote the nerve growth and survival. So they are produced by the nerves, muscles, glands, and astrocytes. Known neurotrophins are the nerve growth factor, brain derived neurotrophic factor, neurotrophin 3, neurotrophin 4, etc. Next, the nerve growth factor. The first neurotrophin to be identified. It promotes the growth and survival of the sympathetic nerves and some sensory nerves. And autocrine survival factor for memory B lymphocytes. Found in high concentration in the saliva. It decreases apoptosis of neurons by acting to the tyrosine kinase A receptors. Brain-derived neurotrophic factor BDNF. It promotes the growth of the peripheral sensory nerves. Acts to the tyrosine kinase A. Neurotrophin 3 promotes growth of cutaneous bacterial receptors. Neurotrophin 4 and 5 acts to the tyrosine kinase B. The exact function is not known. Depending upon the number of processes, depending upon the length of axon, depending upon the function of neurons, depending upon the pattern of dendrites, there are unipolar neurons and pseudopolar neurons have a single process, usually found in invertebrate in ANS and cerebellum. The pseudo-unipolar neurons, axon after originating from soma, splits into the central and peripheral process. Example, dorsal root ganglion cell. According to the arrangement of axon, the bipolar neurons, the two processes arise from the cell body. The bipolar cells of the retina, olfactory receptors, vestibulocochlear nerve ganglia, all these are the examples of the bipolar neurons. The middle process arises from soma in the spinal motor neurons. Golgi type 1 have short axons, the dendrites terminate near the soma, cortical inhibitory neurons. Type 2 axons are long, the cortical motor neurons. Sensory neurons afferent, motor neurons efferent. Dendritic pattern pyramidal cells, they are hippocampal pyramidal neurons. Cortical stellate neurons are the stellate cells. So, this is regarding the hexoplasmic transport and neurotrophins and the classification of neurons depending upon the number of axons, number of dendrites, and depending upon the function. Thank you.